This is a introductory, excuse me, introductory recording about our class project. It is new. We are doing a project that builds on work that was done from 2011 to 2016. And the way the class projects work is we are doing a real experiment. We don't know what the results will be. We may get many mutations, we may get none, and that either of those will be okay. Your mark is never based on whether you find a mutation or not. It's how you describe and discuss the work we did and whether you understand our intentions and what the results mean and what we can and cannot conclude from them and things like that. And the way I work it is that the teams each are responsible for a small part of the work, and then we put it together. I do some of the experiment along with you, so I will be setting up the same kinds of crosses you are. I'll probably do more because I'm in the lab more often than you are, and I have more experience, so I can work a little faster, and that way we'll have larger numbers. So this is a very collaborative project, which means it's important that everybody understand what we're trying to do so that mistakes are not made. If you make a mistake, obviously, you're not going to fail or anything, but we will have to throw that data away. And so it just diminishes the value of the work we do. And gives us smaller numbers to work with. The fewer the flies we test, the less likely it is we're going to find a mutation. Okay. One thing that I don't think I've actually mentioned yet, but it's something to keep in mind as you work with your teams. At the end of the semester, I am going to ask the teams to evaluate the other team members and themselves in terms of their contribution to the group. And that doesn't mean that everyone did exactly the same, but that everyone made a contribution that was of equivalent value. So if you are the person who promises to collect virgins and then never shows up to do that and doesn't let anyone know, you can expect the evaluation from your peers to be lower than if you follow up on things, and that if something happens that prevents you from doing your part of the work, you let someone know so that they can cover for you. You offer to cover them at another time, that you're cooperative, that you contribute something. One of the commonest reasons why students don't like group work is there's always a perception that someone's getting a free ride. Someone is sitting there taking the ideas of the others, getting a grade, and not actually putting in the effort. Now, in my experience in this class, that does not happen very often, but we do want to discourage it. And so there will be a grade out of 10 that will be based on your peer evaluations, how your peers evaluate you, and it will be added to your final paper grade. So instead of being out of 60, the paper will be out of 70. And just if you do the math, you will see that getting a 10 out of 10 on the evaluation will increase the percentage you get on your paper, whether you get a good or a medium grade on your paper. So it matters. And I'll also be checking the self-evaluation. My experience of that is that it's hard to evaluate yourself honestly, and most people in the class give themselves exactly 1 point less than their peers give them. But we have had some very notable examples of people who gave themselves 8 out of 10 and whose peers said, we're giving him 2 and we're only giving him 2 because we don't want to give him 0. So evaluating your own contributions in an objective way, it's challenging, but it's actually a very useful skill, because in many companies, in many types of jobs, you may be asked to submit peer or self-evaluation. I worked in a couple of jobs where I was asked to do a self-evaluation and then came in and discussed it with the people who had evaluated me, and it certainly is a valuable experience. Okay, so I won't blather about that too much because a few minutes has already passed, but I just wanted to be sure that somewhere it's recorded that we're going to do that, so that it won't be a surprise to anybody, and I'll probably mention it in lab a couple of times. Okay, so this is the project. We're working on a gene called DMAP1, which is DNA methylase 1 associated protein. So it encodes a protein that associates with another protein that is known to put methyl groups on DNA molecules, usually C's, but sometimes also A's. And that is associated with regulation of gene expression. So if you methylate DNA, you are usually making it inaccessible to transcription factors. And so if you methylate the promoter of a gene, for example, you are most likely shutting it off. Okay, and so that's the idea. And we're going to use a P element, what we call a local hop. We have a P element strain that has an element only a couple of hundred bases, maybe 300 or 400 bases away from the gene that we're interested in. And so we're going to try to mobilize that element and then collect progeny that have an insert somewhere on chromosome 2, and some of those will be hopefully into the gene that we're interested in. This is referred to as a local hop because most P elements, when they look at where they transpose, where they move to, they move all over, but the vast majority usually move within 10,000 base pairs of their original position, so they tend to move just a little short distance on average, and so that would be, that makes it more likely that we'll find a gene, an insert into the gene that we're interested in. Okay, so this is the map. The P element is very close to a gene called DMAP1. There's another gene beside it, LRT. We won't really talk much about that. And the idea is you're going to make a cross that allows this element to move, and it'll move, you know, in different directions, different places. But what we're trying to achieve is this. The element moving into somewhere, hopefully right into the DMAP1 gene, so that it's interrupted in such a way that it cannot make a functional protein. And it's important to note that we can't make it go here. The only thing we can do is mobilize it and then see where it goes and see if we can find one that's mobilized into this position. Okay, so the good news is that we've been working on this gene for a few years now, and so in the last few offerings from 2011 to 2016, with 1 year off when I was away sick, we have been able to use crossing over in males to select for deletions to one side or to the other of the P element, and we have made 2, and they both remove DMAP1. Our collaborator in Bellingham, Sandra Schultz, Dr. Sandra Schultz, has confirmed with PCR. Her students used PCR to confirm that the DMAP1 gene is missing. In your folder that includes information on this gene, there's papers on the background. There's also a link to the blog post that Dr. Schultz wrote that describes all of this work and a lot of background on DMAP1 and the techniques. I strongly recommend, you don't necessarily have to read every single word of it, but the DMAP1 parts you should at least go through a little bit, because it provides you very important, well-written description of the project, how it started, what we've done so far, and our work is going to build on that. Okay. The problem with the deletions we made is that they do not only remove DMAP1, but they actually delete far up from the original site of the P element, and so at least 27,000 bases away, and maybe even larger. They both seem to be fairly large deletions, and so we don't know if the homozygous lethality, the fact that you can't make that deletion homozygous, whether that's because DMAP1 is deleted or perhaps it's because of some other genes. And so we want to be sure that if we study that lethality, try to figure out what stage and how it works and all that sort of thing, we want to be sure that it's DMAP1 and not one of these other genes. Okay, so the approach that we're going to use is we're going to make a cross that allows the element that's beside DMAP1 to be mobilized. It's non-autonomous. We're going to have to bring a wings clipped element into combination with it, and then collect males where that mobilization is going to be happening, and then collect progeny from the male that has both the wings clipped and the non-autonomous element. Each of the male progeny will have a chromosome 2 that may or may not have an insertion mutation on it. And then we'll make a whole bunch of males that have possibly this mutation. And then we're going to cross each one individually to females that have the deletion that we made in 2014. And then if any of the chromosomes we collect and cross to that deficiency show lethality in combination with that deficiency, then we're going to keep a balanced stock of that and study it further. It will most likely have a p-element inserted into one of the genes in that deficiency interval, and one of those is essential, and one of them maybe more of them will be inserts into DMAP1. And we call this selecting for lethality over a deletion. So that's the name of it, and this is what it means when we say that, selecting for lethality. Or screening over deletion is another way we might say it. Okay. And so this is the idea. If we have a chromosome 2 where a p-element has actually inserted, say, into DMAP1, or maybe one of the other genes that's nearby, that's what we're trying to make. And then when we combine that with the deletion, this white area is the area that's been removed, so these genes are not present. So if you've got a mutated copy on one chromosome and on the homologue the gene is completely missing, then we expect if DMAP1 is required for survival of the flies, then we expect that to be lethal. Suppose DMAP1 isn't required for survival but is required for, say, proper fertility or something, then we would expect that combination to be viable. You'd get flies that had both, but then maybe they couldn't reproduce, or maybe there'd be a visible defect. So we're really going to be looking at the phenotypes that may be associated with this too. But the first part is to just collect ones that are lethal in combination. Okay, so the reason for this, and I usually start with DMAP1, but I realized that I hadn't actually put it there, is that a colleague, as I said, Sandra Schultz, we did our PhDs together, so we've been friends for quite a long time, and we, one of her colleagues was able to isolate a cDNA of this gene, DMAP1, in a parasitic wasp, and it had some interesting features, and the fact that it associates with DNA methylase and that may be associated with transcriptional regulation is very interesting. And so she started looking up what is known about it, and there's almost nothing known in flies, which is really interesting. There's a little bit known in vertebrates, and most of the work is in cell culture. And cultured cells are somewhat abnormal, but at least it has given a suggestion of the kinds of things that this gene may be involved with. And we'll talk about those in a second. This is just a little picture from Flybase that just shows the area of the chromosome where DMAP1 is located, and it just shows you each of these little figures here shows different genes, and the p-element we're going to be using is this one here. Each of these is an insert into the gene. When you look at Flybase, it shows you the locations of all kinds of things. It shows you where cDNA, where transcripts have been identified, and it shows you all the insertions we have, so that if you want to study a gene, you can pick the one closest to your gene and then do what we did last time, which was to try to make a deletion into it. And so this shows you how close it is. It's right in the front end of the gene pointing in the opposite direction, transcribed the opposite way, but it's only a very small, maybe 300 bases away from the DMAP1 gene. So that was our starting point, that gene. And that's, at the time we started this work, that was really all that was known. It was there. And we knew that there were p-elements next to it. We didn't know much about it, but it's had some connections to a number of different kinds of processes. So a connection to gastric cancer in vertebrates, some connections to DNA repair, the maintenance of stem cells, the maintenance of embryonic stem cells, regulation of ATM activity. ATM, ataxia telangiectasia mutagenic. ATM is a protein involved in DNA repair, and ATM mutations are known to be involved in the development of cancer. And if you have a mutation, if you're heterozygous for a mutation in, for example, the ATM protein, you are predisposed to developing cancer in your lifetime. And so these are very interesting things, and it would be kind of cool to be able to study these in flies, because you can sometimes really get at genes more easily in flies than you can in humans or mice, where there's a lot of redundancy. Okay. And maybe what I'll do, I'll stop here and I'll just do another quick one that just takes you through a little bit of what we did in the last few semesters, and then I'll talk a little bit more about our project, but not in detail, because in the actual lab you're going to be designing the screen that's already actually underway, and that way you'll understand it better.